Black-Eyed Susans versus climate change. Who will win? The potential effect of increasing temperatures on black-eyed Susan's growth rate. As climate change emerges as an increasingly critical global issue, the question of how native plant species will respond to shifting environmental conditions remains a central concern in ecological research. According to Croas et al. 2019, the rate of warming currently experienced by many species is unprecedented in evolutionary history, with predictions of 2 to 5 degrees Celsius warming likely to occur this century. Virginia native plants will exhibit varying growth rates and higher than average temperatures caused by increasing global warming conditions. The black-eyed Susan plant, Rudbeckia hirta, is a flowering native plant to Virginia, typically preferring a temperature of around 21 degrees Celsius for optimum growth. The black-eyed Susan plants will have a higher growth rate in higher temperatures than those in lower temperatures. Starting with Miracle-Gro potting mix, we then created a 1-inch indent in the soil for each set of seeds to be added, and placed 2 seeds per pot with 20 pots per treatment group. Each treatment group was given 500 milliliters of water to reach a pre-measured marked water line. Once prepared, each group was transferred to its desired location, the incubator or greenhouse. We watered the flats every 3 days, replenishing the water to the predetermined fill line. The experiment had 3 treatment groups. The first group was in the greenhouse at an average temperature of 23.3 degrees Celsius, where it was exposed to the natural sunlight cycle during the day. The second group was placed in an incubator at a constant temperature of 24 degrees Celsius. The third group was placed in a second incubator at a constant temperature of 26 degrees Celsius. Both Group 2 and Group 3 were on an LED light cycle that turned on at 7 a.m. and turned off at 7 p.m. After 14 days, each treatment group had a 100% germination rate. To collect the growth data, each seedling was removed from the pot and placed in a dehydrator for 24 hours. Overall length and mass, along with shoot and root length and mass, was collected and recorded in an Excel data sheet. The following Excel sheets represent our raw data before analysis. Overall length and mass, along with shoot and root length and mass, were collected in milligrams and millimeters. We felt that the following 3 graphs were most relevant to the plant's physiological response to increased temperatures. When comparing all 3 treatment groups, the first incubator, set at 24 degrees Celsius, had the longest average length. This incubator was set at the plant's optimum temperature found in nature, so the recorded results are most likely due to the consistent LED light exposure and optimum temperature setting. Analyzing the average total mass gives insight into water retention in extreme temperature conditions. More specifically, treatment group 3 and incubator 2 at the highest temperature of 26 degrees Celsius had the largest average mass of 7 milligrams. According to Schulter et al. 2002, root soil interactions are an essential part of global matter cycles as all water and nutrients are taken up. Our root mass data demonstrated increased water uptake at the highest experimental temperature, 26 degrees Celsius. Our findings from this experiment are aligned with both our hypothesis and prediction. As we stated in our hypothesis, varying growth rates were prominent in each treatment group. Also, our prediction was correct that higher growth rates in correlation with our black-eyed Susan mass occurred at higher temperatures. Our data analysis proved root mass was directly related to water collection at higher temperatures. According to Foot et al. In a discussion about a study on black-eyed Susans, its root systems has a high surface area for optimal water absorption. The significance of our analysis demonstrates the Virginia native plant's ability to adjust its physiological processes to alter its range of tolerance in response to potential temperature fluctuations. According to Croas et al. 2019, understanding how temperature dependence of physiological processes is related to climate variation is critical to predict how species will adjust to warmer temperatures.